Today I'm gonna talk about biodegradation of using engineered yeast. The topic of main paper is development of yeast fuel cell biocatalyst for amide conversion into terephthalic acid and ethylene glycol. This is the paper I wrote. Polyethylene terephthalate PET is one of the most abundant plastic globally, with over 56 million tons produced annually for use in food packaging because of easy production and low cost and variety of material. PET is easily produced by esterification of the ethylene glycol and terephthalic acid. However, over the 70 years since the introduction of plastic into every items, plastic waste has become an increasing problem. PET is difficult to recycle because it is inefficiency as it involves high temperature and pressure and often produced by product. Microplastic and nanoplastic accumulate in animals from mollusks to humans. So new methods need to be developed to manage and eliminate and recycle plastic waste. Recently, multiple enzymes capable of degrading PET plastic have been identified and engineered. Peptase and amylase from Ideonella saccharensis depolymerize PET into two building blocks, ethylene glycol and terephthalic acid. So importantly, ethylene glycol (EG) and terephthalic acid (TPA) can be reused for PET synthesis, allowing complete and sustainable PET recycling. In this study, they used Saccharomyces cerevisiae, is known as baker yeast and grass yeast. It is a species utilized widely in bioindustrial fermentation process as a platform to develop a whole cell catalyst expressing the amylase on the cell surface. And cell surface display is a system for attaching and expressing enzyme to the yeast cell surface. So yeast itself acts as an enzyme called whole cell catalyst. An enzyme is immobilized to the cell surface, so it is more stable against temperature and pH than free enzyme. So in this study, they created yeast that converts monohydroxyethyl terephthalic amide into TPA and EG by immobilizing amylase on the surface of Saccharomyces cerevisiae cells. And then they assessed the 6 expression architecture called anchor protein and identified those resulting in efficiency amylase expression on the cells on the yeast cell surface. This is a map of yeast strain construction. And WTC is a promoter, and OST is a signal peptide that allows secretion to the cell surface. And amylase is an enzyme sequence that we want to express. And GFP is green fluorescent protein, and it allows confirmation of the expression location of gene amylase. And display partner called anchor protein. It is a coding sequence of protein that can attach amylase on the cell yeast surface. The anchor protein shown here is AGA2, CCW12, CIS3, CWP2, SCD1, and TIP1. And we will compare these six. In this study, we will compare these six display partners. And PRM is terminator. And they use, there are a lot of methods to make new strain using genetic engineering, but they use the CRISPR-Cas9 system. CRISPR-Cas9 system is bacteria's immune system against impacting bacteriophagy. The specific repeating sequence of bacteria is CRISPR. So when virus invade the bacteria cells, the genetic information of invading virus is recorded between CRISPR sequence. And guide RNA derived from a CRISPR sequence recognize the sequence of virus and bind complementary to it. And Cas9 protein, which is cutting enzyme, recognize a specific sequence of virus and cuts DNA. So it is an immune system by destroying virus genome. So people used this system for genetic engineering. So to do this, you need a toast strain and Cas9 protein for cutting the DNA and guide RNA and donor DNA. So when a plasmid containing guide RNA and donor DNA are expressed in a strain containing Cas9 protein, the target sequence is cut like this, and the donor DNA is introduced at the same time using the repair system, thereby obtaining the desired genetic mutation. So this is fluorescence micrograph for the MFT surface display, and the right picture is GFP. You can measure MFT's total protein abundance and cell surface abundance. And the left picture is con A594, and you can determine the cell boundary. I think you might be wondering about what it is. So it is a labeled cell. It is imaged cell labeled with canavalin A conjugated to Alexa flora, which bind to glycoprotein in the cell wall. And this is MFTase with anchor protein. So you can compare the expression efficiency of anchor protein. And they assay the correction of MFTase displayed at the cell surface by measuring GFP signal at the cell surface relative to total GFP signal by analysis of fluorescence micrograph of a cell expressing MFTase. So this horizontal bar is mean of the replicate, and as you can see, MFTase with AGA2 anchor protein and MFTase with SCD1 anchor protein had the highest display fraction, like this. And the graph below is about the abundance of MFTase at the cell surface. It was calculated by fraction above, and the display protein abundance was more variable for MFTase with AGA2 anchor protein and amylase with CIS3 anchor protein and amylase with a CD1 anchor protein as compared to the other constructs. So it suggests that cells might not display this chimera uniformly. And also they assessed amylase activity. Amylase activity is assayed with the substrate MP and PT. Amylase hydrolysis of MP and PT accurately reflect hydrolysis of MF. Cells expressing amylase were incubated with increasing concentrations of MP and PT, and PNP formation was quantified. This is the result of amylase activity. As you can see, it shows difference in reaction rates and substrate affinity depending on the type of anchor protein. Here, amylase AGA2 anchor protein performed poorly, and amylase with TIP1 anchor protein having had the highest reaction rate. And cell expressing intracellular and did not hydrolysis MP and PT. It demonstrates that MP and PT is hydrolysis by surface displayed amylase. And then recombinant amylase produced in E. coli and secreted by yeast behave similarly to the other display amylase. And they assessed whether the amylase full cell catalyst could hydrolyze its natural substrate MHET. They choose amylase with TIP1 anchor protein full cell catalyst as it performed well in their previous assays. Cells were incubated in the presence of 1 millimole MHET, and TPA formation was measured by HPH after 1 hour and 24 hours of incubation. And the below graph is about the result. In amylase TIP1, TPA was detectable after 60 minutes, as you can see, and after 24 minutes of incubation, all MHET was converted to TPA. In contrast, extracellular amylase, little TPA was detectable even at 12 full hours, and it indicates that amylase hydrolysis was occurring extracellular. So they concluded that full cell catalyst hydrolyze MF. To accurately compare the difference, kinetic parameters were calculated using Michaelis-Menten plot, and turnover number KCAT of amylase with TIP1 anchor protein was 68% of purified amylase and 96% of secreted amylase. And Km value for the displayed enzyme were greater than purified and secreted amylase, indicating that surface display reduced the substrate affinity of amylase. And catalytic efficiency for the whole cell amylase catalyst was also lower than the recombinant or secreted amylase. And this is amylase activity depending on the pH. As you can see, amylase activity was optimal for all the chimera at pH 7.5. At higher pH, the system remained active, but active was reduced by approximately 40 to 50% like this. And MFTase TIP1 anchor protein was the most active, and MFTase with AGA2 anchor protein displayed a lowest activity. This is MFTase activity depending on the temperature. Activity steadily increased and peaked at 45 degrees for all the chimera. At 55 degrees, MFTase activity was lower. Therefore, 45 degrees was optimal temperature, and purified recombinant MFTase also showed optimal activity at 45 degrees. And this is MFTase activity depending on the time. MFTase activity increased over the time like this. But the cell count decreased over the same period of time, suggesting that cell lysis was occurring, and that release of intracellular MFTase could be mechanism by which activity was increasing. To test this hypothesis, they repeat the MFTase assay at the day 12 on the cell pellet and cell supernatant. And they observed the strong activity in the supernatant, representing approximately 70 to 80% of enzymatic activity over the cell suspension. All MFTase activity for intracellular construction was in the cell supernatant, consistent with release due to cell lysis. However, activity in the cell pellet was higher at day 12 than day 0. It indicates the substantial increase in activity for the whole cell catalyst deposit considerable cell lysis. And by contrast, soluble purified MFTase was inactive after 4 days at room temperature, so the whole cell catalyst retained greater activity during prolonged incubation than the purified enzyme. So in this study, they created the yeast MFTase, which can convert MF to TPA, is expressed in Saccharomyces cerevisiae using cell surface display system. At this time, expression efficiency was compared for each type of anchor protein. Anchor protein AGA2, CWP2, CCW12, TIP1, and CIS3, and SCD1. And this is conclusion. A new system for degrading MF, an important byproduct of MF plastic degradation. Using a cell surface display strategy and testing multiple display fusion partners called anchor protein, they demonstrate the production of MFTase. AJ2 was a poor display partner for MFTase because display efficiency was good, but KM value was higher than the others. And TIP1, CWP2, and SCD1 anchor protein was good display partner for MFTase because of sustainable kinetics and display properties. MFTase whole cell catalysts were active for at least 12 days and retain activity up to 45 degrees. So stability So when combined with time and cost saving realized by avoiding enzyme purification, stability gains related to purified soluble amylase. So the yeast surface display is viable route for amylase production. And other studies, in this study, it's about a full cell biocatalyst by displaying peptase, not amylase, on the surface of yeast Pichia pasteurist cells to improve its degradation efficiency. In this study, the amylase whole cell catalyst performed the first step of PET biodegradation, unlike the main paper. The main paper was about the not peptase. It's about the amylase whole cell catalyst perform the second step of PET biodegradation pathway. So this is the reference that I referred. Thank you.